Hello, class, and welcome to Chapter 9, the Muscular System lecture. So the first thing to start off with is thinking about the muscular system and thinking about muscles as a tissue. So if you remember, a tissue has more than one different type of cell in it to actually create some sort of function. So when we think about muscle tissue, there are three different types. You have the skeletal, smooth, and cardiac. And I want you to be able to not only distinguish between the three, I also want you to know some characteristics. We're going to spend a lot of time talking about skeletal muscle, and sort of at the end, we'll just sort of make mention of cardiac and smooth muscle. But when we think about skeletal muscle, it's going to be actually attached to the bones of the skeleton via what are called tendons. And we're actually going to mention there's a couple different structures that help to attach muscle to bone, but skeletal muscle is what's attached to bone, and it's going to have conscious control, meaning it is voluntary. So when you think about talking and you move your mouth or you wiggle your fingers and toes or you throw a ball, that is using skeletal muscle. It is consciously controlled. Cardiac muscle is going to be found in the actual wall of the heart. It's referred to as the myocardium. And whenever we see myo or myos, it's referring to muscle. So the myocardium is the heart muscle, and it makes up most of the wall of the heart. Now, this is responsible for the pumping action to allow for blood flow to happen throughout your body. And we have no control over this. So this would be considered to be involuntary. And smooth muscle is also involuntary. We do not have control over this. So where we're going to be thinking about smooth muscles is areas where we have to propel food. So in our stomach, in our small intestine, large intestine, when we're thinking about our blood vessels and having that constriction and dilation in our pupils, that's where we'll be finding smooth muscle. So make sure you know some characteristics and make sure you can distinguish between the three. Now, skeletal muscle, again, is usually attached to bones, and we actually have it attached to the skin of the face as well. And when we think about the muscular system, skeletal muscle is the organ. And so again, when we think about this organ, it's made up of several different types of tissues. And so the skeletal muscle itself, again, is something to think about, that's the main muscle or the main organ of the muscular system. And when we think about skeletal muscle, again, it has muscle tissue. It also has to be innervated, meaning that you have to have nervous system control in order to make it work. It also will have blood, and it'll have different types of connective tissues actually linking it to the bone. So we'll discuss those more here in just a moment. So when we think about these connective tissue coverings, in this picture here, you can see two of the three. So a fascia is sort of a covering over an entire muscle group. If we were looking at the quadricep muscles, the anterior component of the thigh, they would be wrapped in what is known as a fascia. We also have tendons, and I always try to make mention of this, that ligaments link bone to bone, they're like ligaments, and tendons link muscle to bone. Tendons are more of a cord-like structure when you see it, and again, they're going to be that attachment from muscle to bone. But we also have something as well that's more of a thin sheet-like structure, and it's called an aponeurosis or aponeurosis, depending on who is speaking. So we can see these aponeurosis in the head, as well as we can see this in the abdomen. So when we think about the muscles in the head, we have the frontalis, which would be that frontal muscle. And then we also have an occipitalis as well. And there's this more flat-like structure that's connecting them together. With an aponeurosis, it can link muscle to muscle, or it can link bone to muscle. So again, we can see the aponeurosis in the head, as well as we can also see it in the abdomen that's making up that six-pack that this man has, or female, you can't tell with the muscles. Next little piece that we're going to see are the different types of connective tissue coverings that are going to be surrounding the muscle. Now, what you're going to see is that there is a portion that is going to surround the whole muscle, a piece that surrounds individual fascicles, and then there's a part that is going to be within what are called fascicles. And so I have to really kind of hold up for a moment and kind of describe what a fascicle is really quickly. So this way it makes a little bit more sense. A fascicle is going to be a bundle of muscle fiber. Now, really quickly, I want to make mention of this so that way it makes a little bit more sense. when we think about muscle fiber, that is the equivalent of a muscle cell. So a muscle fiber is a muscle cell. So when we think about a fascicle, it is a bundle of muscle fibers that are held together. If I could spell today, this would be fantastic. And so when we think about these connective tissue coverings, we have a covering that goes over the whole muscle that is known as the epimysium, and that's what we can see here. Now, this more sheet-like structure, that's the fascia that we see. So again, when we look here, we can see that we have the bone, we have the tendon, which is like that cord-like structure, and you can see sort of this white saran wrap, if you will. That is the muscular fascia. And then there's this epimysium that surrounds the whole muscle. Then what's going to happen is inside the epimysium, you see these bundles. And again, these bundles of fibers are fascicles. So around the fascicle, we're going to find the perimysium. And so if you think about epi is meaning above, mys or myo is meaning muscle. Peri means surrounding. And then what we're going to notice is within each individual fascicle, we're going to find what is known as the endomysium. And we can kind of see that in here, the endomysium that's going in. All right, so now we have to get into a little bit more detail. I mentioned before previously that the idea of skeletal muscle is the fact that you'll see these striations. So I always say it kind of looks like corduroy pants. And there's a reason that it looks like this. So what we're actually seeing here is if we were thinking about this muscle fiber, this whole piece here that we're looking at is a skeletal muscle fiber. And again, when we think of skeletal muscle fiber, it's the muscle cell. Now, what we can see is that they are actually composed of what are called myofibrils, and we can see it from this picture here as well. When you get into these individual pieces, so again, this portion here is the fascicle, and within the fascicle, you have individual myofibrils. So these are myofibrils, and this whole thing is a fascicle. And again, you have a bunch of fascicles within this muscle that are going to be here. So we're getting a little bit smaller. So now imagine that we took, again, a picture here of the myofibril and made it a little bit bigger. That's what we're seeing here. So these are the myofibrils now that we're looking at. And if you look at these myofibrils, they are composed of what are known as myofilaments. These filaments are what create some structure. And we can see that there's a little bit of like a bluish color and a little bit of a purplish color here going on. These colors actually represent specific myofilaments. The thin guy that we see here, and we can sort of notice that it's like in this bluish purplish color, that is what we know as actin. And we call this the thin filament. Versus this guy here in the purple that we see is known as myosin. And that is known as the thick. So the purplish color, that really thick look is the myosin. The thinner purpley blue color is the actin. And again, we know them as thick and thin filaments. But when we see these skeletal muscle fibers, again, AKA muscle cells, you can notice that they are very heavily nucleated. So they have more than one nucleus. And again, this should make sense if you think about the function of the nucleus. The nucleus is to hold the DNA. DNA functions to make proteins. You will need a lot of nuclei to sort of make that work. And what you're going to see is around the whole muscle fiber is the sarcolemma. Sarco means flesh. And if you think about lemma, think about it as the cell membrane. That's kind of like a fancy word for it, is cell membrane. And I will ask you on a test to sort of know these pieces. So I would suggest taking a moment, grabbing a piece of paper and writing these down. The sarcolemma is the cell membrane. And within the cell membrane, you have the sarcoplasm. Just like when we think about the cytoplasm, where we find all those organelles and we would find our fluid, a muscle fiber is going to have that too. We just refer to it as a sarcoplasm. And again, when we think of sarco, it refers to flesh. So that's how it gets its name. And again, there are many of these myofibrils that have this actin and myosin. Now, there's some more detail that we have to think about too for a muscle fiber. It's going to be a little bit more specialized. What we're going to see is that they have their own sort of type of modified endoplasmic reticulum. It's called the sarcoplasmic reticulum. So imagine that this is sort of a modified, it's really like a modified smoothie R when we actually see it. But it has a sarcoplasmic reticulum, and we can see this actually right here. This is the sarcoplasmic reticulum. Now what happens is there's all these tubules that are kind of combining together. So we're going to see that we have the sarcoplasmic reticulum in this orangey color. And then we also have what is known as a T-tubule or a transverse tubule right here. And what happens is when we put them together, so we have one side of the sarcoplasmic reticulum, we have the transverse tubule or the T-tubule in the middle, and then you have your other side of the sarcoplasmic reticulum. It forms what we know as a triad. And the reason that this actually matters is because this is going to be very important for calcium. Now, I had made mention before in a few videos that when we talk about ions, ions are extremely important, especially calcium. Calcium is needed for muscle contraction. It is needed for nervous system function. It's also needed for cardiac function and blood clotting, as well as actually digestive system work. So when we start talking about calcium, calcium is an extremely important ion when it comes to skeletal muscle function. So just to kind of overview again, the things that you're seeing, again, skeletal muscle fiber equivalates to muscle cell. They have multiple nuclei in here. Again, the cell membrane is the sarcolemma. The cell cytoplasm is going to be known as the sarcoplasm. And within this skeletal muscle cell, you're going to see these myofilaments, and they're formed into these bundles called myofibrils. Within these myofibrils, we find thick and thin filaments. The thin filaments are the actin, the thick filaments are the myosin. And what will actually happen is they form something called sarcomeres. So from this line to this line, we have what is known as a sarcomere. And this is actually the contractile unit of skeletal muscle. We'll talk more about that here in just a minute. In terms of organelles, again, we have a modified smoothie R, which we call the sarcoplasmic reticulum. And with that, along with what is known as the T-tubule, they form what is called a triad. And this triad is very important for release of calcium. You need calcium in order to have a muscle contraction. All right, now getting into a little bit more detail about this thick and thin filament, this actin and myosin that we talk about going on. The myofibrils have what are known as sarcomeres. And so it's from this end to this end, or from this end to this end, where you see these lines. And if you kind of notice, they make a zigzag. So kind of use that as your hint. This zigzag line is what we know as a Z line. So from Z line to Z line, we have a sarcomere. The sarcomere is the contractile unit of skeletal muscle. And what's going to happen is when skeletal muscle contracts, these are going to shorten. And that's why it kind of looks like corduroy pants. And again, this is what's so important when it comes to the actual shortening of muscle. So within this sarcomere, and I do want you to know, if I was to show you a picture, I want you to be able to identify these parts. So again, this line here is a Z line. This line here is a Z line. The Z line is composed of the thin filament actin. And if we were looking at bands, this lighter area that only has actin is what we call the I band or the light band. So I band is the light band, and that should make sense because light has an I. This other area right here that we can see that we have actin and myosin that ends up being a little bit darker, we call the A band or the dark band. And again, that should make sense because A and that dark has an A. It's the way I always remembered it. So you have the I band, the A band, and then we have those Z lines. And what you're going to notice is right in between the A band, you're going to see like these little buttons, if you will. That's what we call the H zone. And the H zone only has thick filaments in this area. This zone, again, if you look, only has that thick myosin filament. And then we have what we call the M line as well. So this is the band or the zone for the H zone because it only has myosin. And then right with these buttons, we can see that M line going down. So just a couple of things to keep in mind. Again, if I was to ask you a picture and ask you to identify, this whole band is the light band and it only contains actin. This whole band here is the dark band and it composes actin and myosin. And again, if you were looking from this line to this line, we're thinking about the Z lines. And the Z line to Z line makes that sarcomere. Now, why this stuff actually matters is because structure relates to function. So here's a better picture to actually see the skeletal muscle fibers in more detail and start thinking about those myofilaments. So again, now we're thinking about the myofibril. Now we're thinking about looking individually at these myofilaments. So again, the I band is the light band. I and I go together, and it only has actin. The A band is going to have myosin and actin as well. And in figure C, you can see that in a lot more detail, that mixture between actin and myosin. And the H zone is just the center of the A band that only shows you those myosin filaments, versus the M line is the anchor in between that A band that we can see. And remember, from Z line to Z line, the lines that sort of look like these little zigzags, if you will, they're the center of the I band, and they were going to form the sarcomere. And what's going to happen is, again, when skeletal muscle contracts, this is going to get shorter. These are going to be able to slide across each other, and it's going to contract. And then again, when we have relaxation, it elongates back out. So this look of this corduroy pants, again, the structure greatly relates to its function. All right. So when we talk about thick and thin filaments, the thick is what we know as myosin. And if you notice this myosin, it has these heads on it, and those heads are actually quite important. So when we look here, you can see the myosin heads. That's very important to sort of know. And again, it's a very thick protein that we can see. Versus the thin filament is what we refer to as actin. And actin, if you look here, is actually has two components to it. It has what is known as troponin and tropomyosin. And those little proteins on there are actually needed when it comes to muscle contraction as well. They help to actually prevent any sort of cross-bridging from taking place and preventing a contraction from happening in certain areas when these pieces are inactivated. So this movement here, this thin filament, again, has two components, this troponin and tropomyosin to it. And what will happen is when the muscles are actually in contraction mode, it needs calcium to change the look of this little guy here, that troponin tropomyosin complex. Yes, it has a funny shape to it. So we're going to ignore that for right now, but it has a funny shape. And again, when calcium combines, it actually allows for the myosin heads to come in and attach to the actin. But if it's just in a relaxed stage, although they're closely associated, they're not actually touching. Now, the big thing to keep in mind is, again, when we think about skeletal muscle, we have to think about the actual contraction taking place. And with contraction, there is a lot of stages and a lot of kind of mechanisms to make this work. It's not a very easy kind of thing to keep in mind. But what's going to happen is when you actually have muscle contraction, It's the movement of those myofibrils. And again, it's actin and myosin actually sliding past each other. This is why we call it the sliding filament model. And what will happen is the sarcomeres are actually shortening. So as the muscle fiber shortens, it actually pulls on those attachment points. Now, when we think about muscle, we're thinking about skeletal muscle that is voluntary. So we have some conscious control to make these guys move. But you have to keep in mind that muscle just doesn't do it on its own. It has to be innervated, meaning that a nervous system response has to happen in order to send out sort of this message, if you will. So we can want to move, but unless we have some nervous system control, it's not going to happen. So what has to happen here is there has to be communication between a neuron and a muscle. And we call this sort of synapse, if you will, the neuromuscular junction. So it's a interaction between a neuron and a muscle fiber. And so when we look at this picture here, this is also going to be important to talk about in terms of the nervous system. So in the next chapter, we're going to be talking about the nervous system function. And what you're seeing here is this is called a motor neuron. And the part of the motor neuron that we're seeing is referred to as the axon. Basically, this chemical message is being sent down. And when it's being sent down, it hits these little bulbs right here. And in this bulb, you're going to see that there is a chemical messenger, if you will. This is known as a neurotransmitter. And that neurotransmitter gets released and it binds on to the skeletal muscle fiber. So if we want to have muscle contraction happen, we have to have proper nervous system responses in order to have this innervation. So a lot of times when we talk about muscles, and even if you're looking at your textbook, you'll see that when we think about muscles, we'll mention their origin insertion as well as their innervation, meaning what nerve basically allows for that movement to take place. If you were looking in the arm, is it going to be the musculocutaneous nerve? Are we looking at the axillary nerve? Are we looking at the radial nerve? There's innervation by specific nerves that allow for that motion and movement to take place. So again, this is called the neuromuscular junction. And what you also will see is that the nerve itself and the muscle don't actually touch each other. There's this little cleft right here called the synaptic cleft. And again, that allows for that release of the neurotransmitter to come in and actually bind onto those receptors. But this is the site where there's this interaction happening. And it's from a motor neuron to a muscle fiber. That's actually important to know because it's going to help you in the next chapter. Not that we have to necessarily know at this moment, but again, it should just kind of make sense. And skeletal muscles only fire when they're stimulated. So if you had issues with motor neuron damage, you're going to see problems with loss of function. So that is something to keep in mind as well. And so make sure you sort of know this. This will come back here in a few days. All right, now, like I said, if we look at this motor neuron, it's got this axon, and at the bottom of these axons, you see these little bubbles, these little terminals. That's what we're looking at here, is the axon, what are called terminals, or sometimes they're called little synaptic buttons. They have different names for them, but we'll call them axon terminals. In these axon terminals, we see these little vesicles that are full of a neurotransmitter. For skeletal muscle contraction, the neurotransmitter is referred to as acetylcholine. Now, I will not ask you to spell this, but I do want you to know that sometimes we shorten it. As biologists, we're a little bit lazy and we don't like to spell everything out. Plus, I'm pretty terrible at spelling. So acetylcholine usually is referred to as ACH. So if you see me write this, that's what we're referring to. But this nerve impulse is carried from the little synaptic terminal, or this axon terminal, into that cleft. And again, that neurotransmitter binds onto the actual muscle fibers. There's receptors on the muscle fiber. And what it's doing is it's causing a change in the membrane. So what ends up happening here is the outside of the membrane is really positive. The inside of the membrane is really negative. And when this happens, when this occurs, it generates what is known as an action potential. And it changes this sort of permeability. It makes it a little bit different. And so again, in order to make this happen, you're stimulating an action potential that's now going to lead down into the muscle. And when this happens, when they get this action potential, it causes a release of calcium to lead to muscle contraction. So that's the ultimate goal, is that we want this action potential to happen. We want to release calcium, and that calcium release will lead to muscle contraction. If you do not have calcium, you do not have a contraction. So something to keep in mind. No calcium, no muscle contraction. This can also be an issue as well that you can see in some sort of pathological disease. All right, so speaking of diseases, I like to mention myasthenia gravis. Myasthenia gravis is an autoimmune disorder. And what happens here is whenever you hear autoimmune disorder, it's your body cells attacking your cells. It's not attacking something foreign, it's actually attacking your self cells. So we have antibodies that are actually attacking these acetylcholine receptors. And when they attack these acetylcholine receptors, it leads to muscle weakness and muscle fatigue. And it's strong weakness and strong fatigue. It's not one of those where it kind of slowly progresses. The person can go from being fine one day to like not walking the next, or having like where they can't pick something up. And it's very scary. So what happens here is, again, they have these acetylcholine receptors that are not able to actually allow for that transmission. They're not binding onto those receptors properly. And again, therefore, you don't get that proper action potential, and you don't get that proper muscle contraction. So the people that are usually afflicted have about 1/3 the normal number. So again, it's not the fact that they're completely wiped out, but it makes it to where there's significant weakness and fatigue going on. So what they actually have to do is they have a drug that inhibits what is called acetylcholine esterase. It's an enzyme that breaks down the acetylcholine. And so basically they're inhibiting that because they just want to have as much acetylcholine as possible, as well as they're taking autoimmune or immune suppression drugs as well to just sort of block those antibodies from being made. Now, when we think about muscle contraction, again, as I said before, you have to have this nervous system control. You have to generate that action potential. And when we generate that action potential, it allows for a release of calcium. Where does it release from the calcium, or where does that calcium get released from? It's that modified sarcoplasmic reticulum. So again, that smoothie R that normally is modified, it's called the sarcoplasmic reticulum. That's where it releases that calcium. So it basically gets that action potential, it feels it, and then again, it's able to start releasing calcium. Why calcium then becomes important is it's going to change that actin filament. So as I said before, actin has two components to it, a troponin and a tropomyosin piece. When calcium gets released, it changes those binding sites. And when it changes those binding sites, it allows for those myosin heads to actually interact with actin. So this is going to be sort of how I'm going to ask you questions for your test and for some upcoming pieces. I want you to understand the stimulation idea of what's taking place. So again, you're going to have some sort of innervation from a nerve. It hits that muscle fiber. That action potential then is going to move to that sarcoplasmic reticulum. When it does, it releases calcium. And when it releases calcium to the cytosol, calcium ions bind onto troponin and they change its shape. So normally it's blocking those active sites, but basically what happens is that troponin and tropomyosin are in a certain shape that allows for this basically inhibition to take place. So that change in troponin alters the position of tropomyosin as well. And now those binding sites are open and they're exposed. So now for binding wise, those myosin heads can actually bind onto actin. And when this happens, it forms what we know as a cross bridge. So I hope this makes a little bit of sense. What I'm going to do for you guys right now is I'm going to stop the video. I want you guys to just watch this and kind of play it back over. And the next piece that we're going to do is we're going to actually start talking about the true cross bridging that happens in that muscle contraction. I hope you have a wonderful day, and I'll see you guys later. Bye.